It's working.
Working? All right,
Yeah.
let's just roll with it then. Okay. Yeah?
Yeah, it's working.
All right. Are you recording?
Yeah.
Okay, cool. You guys ready to get started?
Yes, I'm
All
excited.
right. Cool. So let's introduce ourselves. That's me. I'm Greg. I am the training manager at Tobii Pro. I've been with Tobii Pro for 3 years now. In fact, on the 20th of this month will be my 3-year anniversary. I started with Tobii Pro as a technical support specialist, as I mentioned earlier, so I got a lot of good experience troubleshooting equipment, like I did this morning, and just kind of learning it from the inside out, which was a really good experience for me. I've also have experience working with our Tobii Pro Insight Research Services team. This is our in-house research services team, which is typically contracted by our commercial customers to help with the eye tracking research. You can sort of hire them on an a la carte basis, or to help with project design or data collection or reporting, or have them run the whole study for you. And I've got some experience working with them and going out in the field and collecting data, and that's all been very valuable for me. My formal education is in marketing. I got my degree from Old Dominion University in Virginia. And all of that has led me to where I am today, where I travel all over North America, visiting labs like this, working with customers who've recently purchased eye tracking, and getting to see it in real labs across the country, which is really exciting for me. And that's it. That's basically me. Any questions?
How many institutions have you visited to help approximately? There's no.
I would say probably 70 to 100.
So what type of programs usually?
Yeah.
Well, you say they're all, but
Yeah.
mainly.
It could be anything, and that was like I'm at a UX lab at some major brand one day, and then I'm in the psych department at a university the next day. So it varies a lot. Our largest customer base is definitely in the academic segment, in labs like this.
So it seems like it's increasing, the interest.
Yes, definitely, and it's becoming.
That's worthwhile, that means that.
Yeah, definitely. Interest is increasing and cost is coming down, so that is helping the adoption of eye tracking, both in research fields like this, but even in like consumer electronics. I mean, you're gonna have an eye tracker in your house within 5 years, I'm pretty sure. If the Tobii Tech division has their way, you will. Cool. So I would love to go around the room and have everybody introduce themselves. By the way, do you guys all know each other and work together
Yes.
regularly? Okay.
Yes.
So you'll just be introducing yourself to me then. But I'd love to hear a little bit about the type of research that you do, the goals that you have for eye tracking, and if there's anything that you hope to get out of today's training specifically. Let's address it.
Judy, do you want to start?
Yeah.
Because you're the most close.
Yeah, so my name is Judy, and that's Chinese name, English name is Judy, so it sounds the same.
Okay.
And I came from China, and this is my
Sorry, was it Trudy or Judy?
Judy.
J-U.
J.
yeah.
Okay.
Yeah.
Awesome.
So this is my 4th year here as a PhD student, so I'm about to leave, hopefully, graduate. And so I'm interested in media psychology and how people process online media, and specifically I just proposed a study about how people process online comments, whether there is civility and incivility in the comments, and whether that affect people's processing and emotional responses toward the message. So in terms of eye tracking, I'm interested in whether I can track people's gaze pattern on sentences. So I'm interested in how precise the eye tracking can be, and whether I can see at what point people is looking at which part of a sentence.
Okay.
Yeah, something like that. Yeah.
Sounds good. Yeah, we'll definitely look at that today, and you'll certainly get a sense about how accurate the eye tracker is. Whether or not you'll be able to determine exactly what line or what word the participant is reading will depend a little bit on how much control you have over the actual stimulus design. Because it's only going to be but so accurate. So if the text is very small, then it's unlikely that you'll be able to determine exactly what word they're reading. But if you have the ability to space the lines a little bit further and things like that, then we can work on it. So,
Yeah.
Do you decide a reasonable interest in the program?
yes, yes. Yep. Cool.
It's about me.
Awesome. Who's next?
Okay, I'm Cici, come from China, and I'm kind of newbie in this field.
Okay.
I'm first year doctoral student, so I want to know everything about this.
Okay, great. Yeah, we'll walk through the whole workflow. I mean, there are lots of different ways you can analyze eye tracking data depending on what the research question is, and we'll kind of give you a sense of all the possibilities today. So that'll be good. Yeah.
My name's Courtney. I'm a first year doctoral student as well. Same boat as Cece. This is my first dabble in this.
Okay.
But I'm definitely interested in message creation, in particular with crisis and risk communication.
Okay. Awesome. Sounds good.
Yeah, so I'm a first year doctoral student as well. My name's Sierra, and I'm interested in health communication and like the creation of messages to change people's health behaviors and their intentions to change their health behavior. So newbie to this as well as far as eye tracking.
So program is 3 years, so all the students will be within that range.
Okay. Yeah.
I mean, except Julie.
Yeah.
My name's Namya, and I'm from South Korea, and I am I'm a first year technically, but it's my third semester, so like that. And my research focuses on how media and individual differences interact in someone's like opinion formation, and I'm interested in how people perceive risk in science and health messages, and I am interested in eye tracking because I'm interested in visual imagery like photos, and for my thesis I'm a graphic designer too, so I made infographics, and for my thesis I did not talk about like attention and stuff as much, but I'm interested in how like people process visual imagery and like what kind of elements in the infographics or photos affect people's risk perception. I don't know. Yeah,
Yeah, no,
something
that makes
along
sense.
that line.
Yeah. So as is the case with eye tracking, as is often the case with eye tracking, and with some of the research questions you guys have proposed today, it'll take a combination of eye tracking and then other methodologies as well, right? So like surveys and interviews and that type of thing. So I'll count on you guys to help me with that side of it, and I'll help you with the eye tracking side as we put together our experiment today. Yeah, cool.
So my name is Hong Kyung Lee, and assistant here, and I started working here 2014.
Okay.
And before that, I was at UPenn, University of Pennsylvania,
Okay.
for about 5 years. And before that, I was in Indiana.
I have a
as a
colleague
grad
who
student.
graduated from university.
Yeah, and then when I was at Indiana, I was actually exposed to eye tracking.
Okay.
I did a little study with the MRI. I did the EEG a little bit, and then physio is my main method I'm employing.
Okay.
And I actually haven't collected any data with the eye tracker because there has been some criticism about eye tracking compared to its cost, right? So
Yeah.
you need to find a system, but like the amount of research question you can answer is kind of narrow, and then
Okay.
it's hard to publish. These days it's becoming more popular,
Okay.
and then also it was part of my interest.
Yeah.
So
That sounds good.
since there is opportunity.
So with the physio thing, one thing with Tobii Pro Lab, which is the newer software that we're releasing, is that we've designed it so that it will be able to support capturing other biometric data streams as well.
Cool.
And right now, we already have a full integration with the Shimmer GSR device. So that's something to consider,
In the future.
in the future. Yeah, I mean, Tobii Pro Lab will make it really easy to combine those data streams. Yeah, so cool. Well, that sounds good. Yeah, I will. At some point, I'll collect all of your email addresses today. Probably before we go to lunch, I'll make sure you sign in on my computer before we leave or something, so that I can send you a follow-up email that includes lots of useful reference material, YouTube videos, white papers, webinars, things like that. What is this? Okay. And so, I may make reference to some of those materials as we work together today. So, any other questions before we get started? No? Cool. All right, so we're going to start with learning a little bit about the human visual system, how it relates to eye tracking. So before we even talk about any eye tracking hardware or software, we're going to learn some principles and foundational material. So And the human visual system is made up of two subsystems here. We have the optical system and the perceptual system. The optical system is our eyes. It's kind of like the hardware that looks out into the world and gathers information about light and color and texture. And then that information is passed to our perceptual system, specifically the visual cortex in our brain, where that information is processed and then converted into the image that we see with our mind's eye. So let's focus on the optical system here, our eyes. And I'm going to talk about a few of the most important features of our eye. First one is going to be the cornea. Cornea is the outermost surface of the eye. It's a clear protective lens on the outer surface of the eye, and it's very important to eye tracking, in fact, and we'll come back to it. So just for now, I want you to know that the cornea is the outermost surface of the eye and that it's going to become very important as we work together today. Next we have the pupil, which is the aperture through which light enters and exits your eye. And you guys know that the pupil, when you look at somebody's eye, is that little black circle in the middle, which is actually a black hole, in fact, and it's the hole that light enters and exits the eye through. The size of it is controlled by the iris, but the iris is not so important. It's just the pupil itself is actually going to become extremely important to us today as we work together as well. So as you're looking at this image, the most important things here are going to be the cornea and the pupil. So keep those in mind. Now I want to talk about the retina, which is a sensitive tissue at the back of the eye that receives those images from our visual world and then send the signals to the brain about what is being seen. And our retina is made up of two different types of cells. We have rod cells and cone cells. Rod cells are very sensitive to light and motion. However, they're very insensitive to colors and details. So cone cells, on the other hand, can sense all of the colors and the intricate details in our visual environment. There's a very interesting part of our retina called the fovea. It's right here, it's right by the optic nerve, and what's interesting about the fovea is that it is very densely packed with cone cells. In fact, most of the cone cells in our retina are here in the fovea within our retina. Now, in order for those cells to receive any images whatsoever, we first must have a light source. So here we have our light source there in the top left. That light will reflect off of our visual environment and then enter our eye, passing through the cornea, the pupil, the lens, until finally that light is received by the retina. And the fovea, now circled in yellow, where all of those cone cells are, could also be described as the origin of our visual axis. So if our visual axis starts here at the fovea, this horizontal dotted blue line is our visual axis, and it will end on whatever we are looking directly at. Make sense so far? Awesome. So I would like then for you to imagine that you've entered the grocery store and approached the shampoo shelf. So just get a mental picture in your mind of what the shampoo shelf looks like. Everybody got it? I bet you're picturing something like this, right? This is what the shampoo shelf looks like. Yours might be in English instead of Swedish, but it looks something like this. And you've got, or I don't even know if that's Swedish, but anyway, you've got this nice crystal clear HD image of all of the shampoo on the shelf. And this is certainly what we perceive the shampoo shelf to look like when we approach it in the grocery store. However, if we were to take a snapshot of our visual field during one instant in time, our visual field would look a lot more like this. So here we have in the outer regions of our visual field, that's called our peripheral vision. Things are quite blurry. There's actually a lot less color out here as well. So, we may be able to tell that there's like this light green fuzzy blob up there in the top left corner of our visual field, but that's about all we can say, is that there's this blob of light green fuzziness there. As we move closer to the center of our visual field, we enter what's called the parafoveal region. So things start to become a little bit clearer here. Maybe now we can see that the green blob is made up of individual components, but we still can't sense all of the detail there. It's not until we get to the very center of our visual field, and that's called our foveal region of vision. It corresponds directly with the fovea in our retina, where all those cone cells are, and this is where we are able to sense all of the colors and intricate details of our visual environment. So in this case, now we can read the brands, we can read the ingredients, we can read the pricing, and we can really process the visual information that is required to make a purchasing decision in this case. So almost all of our visual processing comes from this foveal region of our visual field. Now here's another representation of our visual field on the right there. It's about 200 degrees in total. but our foveal region of vision is very, very narrow. It's only about 1 to 2 degrees in the center of our visual field. So we as humans have a very laser-like focus at any one given time. And there's an exercise that every eye tracking researcher has to do at least once, so if you'll all do it with me. Stick your thumb up at arm's length and look at your thumbnail. The size of your thumbnail at arm's length is about the size of your foveal region of vision. So if you wanted to look at anything outside of your thumbnail, say just your thumb knuckle, you'd have to physically move your eyes in order to look at your thumbnail, then your thumb knuckle, then your thumbnail, then your thumb knuckle. And you can feel that your eyes have to physically move in order to process the difference between those two things that are really quite close to one another. And that gives you a really good sense of just how narrow and laser-like our focus really is. And when we collect eye tracking data and report eye tracking data, it is that foveal region of vision that we are reporting. That's what the eye tracker is able to do, is essentially calculate where the eyes are oriented and what is expected to be within that foveal region of vision. Any questions? I'll take a drink of water between every set of slides here. Okay. Well, moving on then, we have eye movements, and there are lots of different types of eye movements. So we're talking about the patterns with which our eyes actually move around in order to process our visual environment. Lots of different types of eye movements that exist, but we will focus on mainly 2 today. And the first one is called a fixation. So a fixation is the eye movement that takes place when we stop to see something. So as we're looking around with our very laser-like focus, if I'm stopped to look at my computer screen here, I'm now fixated on my computer screen. And we'll typically have about 3 to 4 fixations every second. So our eyes are constantly shifting and moving around in order to process different things and perceive our environment. So about 3 to 4 fixations every second, each one lasting anywhere from 80 to 600 milliseconds. And these fixations do reveal processing and interest in something. So when we are fixated on something, there is some level of processing and or interest going on. Whether that's a good thing or a bad thing depends on the context and sort of the research question, but certainly there's some level of cognitive processing taking place when we are fixated on something. And when we look at eye tracking data, our fixations will be represented by the circles that you see here. And if that doesn't make a whole lot of sense right now, it certainly will once you see that overlaid onto an actual stimulus.
So size is actually corresponding to how long they stay?
Correct, yep. The size of the circle indicates fixation duration. Yep. So the other eye movement that we'll analyze today is called a saccade. And a saccade is the eye movement that takes place in between those fixations. It's the zooming from feature to feature. So again, as our eyes are like constantly shifting around and fixating on different things, for example, if I'm looking at my screen now, I'm fixated on that, and then I want to look at my water bottle, I'd have a very quick eye movement, and now I'm fixated on the water bottle. And that very quick motion between the 2 fixations is the saccade. They happen very, very quickly, usually between 20 and 40 milliseconds in duration. And because our eyes are moving so quickly, our vision is largely suppressed. It's kind of a big blur when we're moving from fixation to fixation. So we're not processing much visual information during a fixation. However, the patterns of these eye movements can be very revealing and oftentimes reveal comprehension issues. So I think the best example of that is probably in reading. If you can imagine somebody who's reading a passage of text that they understand, you'll see their fixations and saccades will be systematically left to right, line by line. And if they come across a word that they don't understand, you might see the saccades will start to regress or go up or maybe start flying all over the page as they look for context clues to better define that word. So the patterns of these eye movements, saccades, can be very revealing.
So saccades is like a path of your eye?
Exactly, yeah. It's it has a direction and a velocity, absolutely. When we look at saccades in eye tracking data, they'll be represented by the lines that you see here connecting the fixations. So, here we have then an image that kind of brings all of these concepts together for us. We have a fixation on the horse's face in this image, and remember that's the area of our foveal vision where all those cone cells allow us to see color and detail. If we then wanted to fixate on the horse's harness, we'd have a very quick saccade, and now a fixation on the horse's harness. And then another quick saccade and fixation to the carriage, another quick saccade and fixation to the building behind there, And these 4 fixations and 3 saccades would probably take place in the span of about 1 second if you were to present this image to a participant. It'd be very quick, like, and after 1 second, they've had 4 fixations and 3 saccades, and now they've processed this image up to that point. So any questions on those, fixations and saccades?
So given that, how much data is collected
Yes.
for one, like so if it is a 30-second ad,
Yes.
like, what is the size of data? So can you change the sampling rate? What? I don't know how it's.
The sampling rate is always, with your eye tracker, it's 60 hertz. So your eye tracker collects 60 raw gaze data samples every second. And then we'll filter the raw gaze data into fixations and saccades, which I'll talk about in a second, how we actually do that. But yeah, as far as how large the file sizes get, certainly with a higher sampling rate, file sizes become a little bit larger, but the main consideration there really is the stimulus that you're presenting. If you're presenting, images to all participants, then there's only one original source image file in the project files. Or video, same thing, one original source video file that you present to all participants. But then if you do something like a website where everybody has a very unique experience and creates a unique file for each participant, that's when file sizes start to get a bit larger. If you capture a user camera, files get larger. So